Okay, this video will feature the endospore stain. I don't know what happened to the video I recorded in lab. I just can't find it. I can't go back to lab because we're on shutdown. So briefly, I'll introduce what an endospore stain is. An endospore is a differential stain that is designed to highlight the structure of an endospore. Hopefully you remember from lecture that there are only some kinds of bacteria that can form endospores. Think about those type of bacteria, think about what kind of cell wall they have, what they might look like using a different differential stain, like a Gram stain. And think about some of the common genera we discussed that cause human disease and can also form endospores. The endospore stain starts out the same way as almost all stains do. You take your liquid culture, so just pretend I'm taking my liquid culture, sterilize my loop, dip into my liquid culture, and spread it across my slide. Once I've done that, I'm ready to move on to the actual staining steps. And so that's where the video will pick up.
Okay, so I did not show you, but I actually did heat fix the slide. Remember, we always want to heat fix. And so now we'll do the endospore stain. So we put the slide on the heater, just like before. And this time we use malachite green. So malachite green, let me show you. Malachite green is the stain we will use to stain the endospores. We have to do this under heat because, as you might remember, endospores are very, very robust. So I'm going to cover my smear. I'll keep an eye on this. I should be heating it for 3 to 5 minutes, and I just don't want to let it go dry. So when we're done, after that 3 to 5 minutes, we will head back to the micro lab and rinse using water. Okay, I'm back in the lab with the endospore stain. So I am going to rinse off all of the excess stain using water. It's a real pretty, like, kind of greenish blue. So there's a lot on there, so I'm just going to rinse, rinse, rinse. Now that I have rinsed, I am going to have to add a counter stain. So with the endospore process, that malachite green hopefully got all inside of those endospores, but anything that's not an endospore, when I'm washing away, should have lost the malachite green. So I still want to counter stain any cells that might be in there that don't have an endospore, or the vegetative part perhaps that's still surrounding an endospore. So I'm going to use safranin as my counter stain. So I will cover the slide in safranin, allow it to sit for 2 minutes, rinse with water, and then blot dry. So I won't show you all of those steps because you've seen them multiple times before. But again, I'm adding the safranin. I'm going to let it sit for 2 minutes. Then I'm going to rinse, blot dry, and visualize under the microscope.
So now I'm going to show you two different images. The first image is from the stain that I recorded this morning. Unfortunately, my culture of bacillus was only about 48 hours old, and I had a really difficult time finding spores in that culture. Ideally, we would like to use a bacillus culture that is 72 hours old or older in order to see the endospores. So I want you to think about why it is I would want to use an older culture, 72 hours or older, in order to really see the endospores. This second image is an endospore stain where you can clearly see the green endospores and the vegetative pink safranin stained bacillus. This is an image prepared by Manny. Manny is the teaching assistant for the 4 p.m. laboratory section. His master's thesis project involves working with bacillus, and so this is an endospore stain that Manny did. So you can hopefully really see the contrast between the endospores and the vegetative cells. I wish I had been able to show you that today using the image I took, but again, unfortunately, I am not able to go back to campus until the shelter-in-place order is lifted. So hopefully Manny's endospore stain will be sufficient to show you what you should be seeing.